The MIT Edgerton Center presents Translation, to be used with the MIT Edgerton Center DNA, RNA, protein, and tRNA sets. The ribosome uses transfer RNA to read the messenger RNA and make a protein. This process is called translation. Transfer RNA, or tRNA molecules, carry amino acids to the ribosome. Each tRNA carries one specific amino acid. When a tRNA has an amino acid attached, it is called a loaded tRNA. Load each of your tRNA with the amino acid on the sticker. After leaving the nucleus, the mRNA travels to a ribosome. The mRNA always starts with A, U, G. The A should start with a phosphate group. The AUG of the mRNA strand is placed on the start section of the ribosome. Place the loaded MET AUG tRNA in the P site of the ribosome. MET is always the first amino acid in every protein chain. The tRNA should bind to the mRNA with all nucleotides pairing correctly. Place the loaded pro-CCC tRNA in the A site of the ribosome. It should bind to the mRNA with all nucleotides pairing correctly. Remove the MET amino acid from its tRNA and attach it to the pro amino acid to make a peptide bond, as shown with the arrow. Slide the mRNA over by 3 nucleotides. Detach the empty tRNA from the mRNA by pinching and move it into the E site. E is for exit. The tRNA is then released to be reloaded and used again. Place the loaded SER UCU tRNA in the A site of the ribosome. It should bind to the mRNA with all nucleotides pairing correctly. Remove the MET pro chain from its tRNA and attach it to the SER amino acid as shown with the arrow. Slide the mRNA over by 3 nucleotides. Detach the empty tRNA from the mRNA by pinching and move it into the E site. The tRNA is released to be reloaded. Continue adding the correct tRNA one by one to the A site, moving the protein chain to the right to add the next amino acid. Remember to slide the mRNA over and detach the empty tRNA. The order of the amino acids is determined by the order of the mRNA nucleotides. Every group of 3 nucleotides is a codon and codes for a specific amino acid. The mRNA codon must pair exactly with the tRNA anticodon for the amino acid to be added to the protein. The stop tRNA is actually not a tRNA molecule. It is a protein molecule called a release factor. Place the release factor in the A site of the ribosome. It should bind to the mRNA with all nucleotides pairing correctly. When the release factor binds to the stop codon, all the molecules are released from the ribosome. Remove the protein chain from the tRNA. Remove the empty tRNA, the release factor, and the mRNA from the ribosome. The protein created here is a beta-mutated channel protein chain. Translation completes the central dogma of molecular biology. More resources at edgerton.mit.edu/DNA-proteins-sets.